Hi, my name is Chenchen. I'm a research scientist in Mike Snyder's group here at Stanford. And thanks for the invitation to let me speak about our experience using Illumina spatial technologies to understand the biology of human intestine. So here's one disclaimer slide. And in Stanford, our tissue mapping center is part of the Human Biomolecule Atlas program. And our goal is really to map the complexity of the small and large intestine. And it's a really effort from our group. Here I list some of the key contributors here on the left. And our goal is to use different assays to understand what's the tissue structure and what are the functional units of the different sites across the intestine. Obviously, this has huge implications for building a reference map and also can contribute how we can understand the disease states. And here's one map showing the full thickness of human duodenum sample. And that's an FFPE block we have. And you see the mucosa area here on the left, and then the submucosa and the muscle layer. So we are doing spatial profile on these samples. And clearly, the focus is really the mucosa with all the immune cells. But the other interesting parts are the muscle and the submucosa. And that's the sample I'll be talking talking through this presentation. And in the phase 1 of the HARMAP project, we have used the Codex technology. It is a spatial proteomics methods that can target about 50 antibodies using different cell type markers to understand what are the cell types and how they are structured together. And we've worked out a very nice spatial map for both the small intestine and large intestine. Here you can see. And when you cluster the cell types together on the spatial map, then we can draw out these neighborhoods and then how the neighborhoods, they form a large-scale communities and what are the tissue units. So you can even see the tissue organization of how the cells, they are organized for the intestine tissue. And this is published 2 years ago. So when I started in Stanford, my goal is to really establish spatial transdermics for mapping tissue cell types and states. And the reason we choose RNA over protein is that RNA, we would have more targets than protein targets. So the protein is only 50 to 60 targets, and RNA, where you can easily go 500 or even a whole transcriptome. And also the choice for the targets is much more flexible. We are not limited to antibody availability. Virtually, we can target all the targets in the transcriptome. And by looking at RNA, it also allows to look at gene regulation. And of course, we don't stop doing the spatial proteomics. So that means in addition to proteomics, spatial proteomics data, we are also looking at the RNA layer, and then we can integrate these two layers together for a multi-omic integration. So my goal is to here is to evaluate Illumina's spatial technology for cell atlasing purpose in the context of human intestine, and that's the whole project is about here. And so how does it work? So we have intestine samples and that we send to Illumina for profiling, and these are OCT blocks, preserved fresh frozen OCT blocks, and my colleague Mohan coordinated this effort together with Illumina. Here you can see on the left we have 5 blocks, so 3 of them are healthy tissues for control, and 2 are disease samples as polyps. And you see on the right, here's the RNA quality and integrity for QC measurements. So it's pretty standard. All of our blocks are of high quality. So the ring score are all over 8 and DV200 values are all above 95. And here's the workflow of the Illumina ST assay. And it's pretty standard and compared to the other spatial technologies out there. So we have the tissue preparation and then the image of the morphology and then the tissue polymerization, and it does the reverse transcription, and then we generate the cDNA and the library prep and the sequence. So because the flow cell, the tissue mounting area is fairly large, so we were able to put 4 samples on one flow cell. So for our 5 blocks in total, we sequenced 3 flow cells. And here you can see the QC for the libraries here. So you see the standard QC here on the right. And another QC, once we got back the data, is to look at really the transfer coverage over the tissue. sometimes we experience in the past that some immune cells, and especially the laminopropia area, it is very difficult to get a very good measurements. But here you can see the transcripts per and 10 micrometers in square. And you see for both the healthy tissue on the left and the polyp on the right, the coverage it looks beautiful. So we have a very good coverage for the whole tissue. And also for the mucosa area where the most immune cells and all the absorption happens, we have a very high coverage in terms of transcripts. And the other thing that's important for spatial assay is really to get the cell boundaries. And to this end, Illumina has developed a software to do the registration of the morphology image and use this morphology image for the cell segmentation purpose. So you can see the registration on the left. So it is registered to our tissue, and then the software does this nuclei segmentation based on the HE signal. And you can see example on the mucosa layer, and we can further zoom in. Obviously, the more accurate the cell segmentation is, the quantification will also be better for the downstream analysis. So we did kind of eyeballing to verify that cell segmentation makes sense. You can see the nuclei here captured and the mucosa, and you can further zoom in for the mucosa layer. It's doing a very good job. You can tell for the mucosa that we are really capturing the nuclei, and by expanding only 5 micrometers, I think we are on the more conservative side. And for the muscle layer, because the muscle cells are irregularly shaped, so we are not really capturing the cell boundaries, but the nuclear boundaries, it works fine, but mostly we are losing a little bit of signal here. It is a very I have to acknowledge it is a very heterogeneous tissue type here with a human intestine, but I think the cell segmentation is as good as we have got so far with all the other technologies. And here's the overview of the data set. You can see on the left is the diseased polyp samples. On the right, starting with the B, are the healthy samples. Overall, we are getting at least 500 transcripts per sample. So this, compared to the other assays that we tried, it's much higher in terms of sensitivity, and we are getting at least 400 or 300 genes per cell. So this is a pretty impressive number for a untargeted approach. And here's the overview of the whole data set again. So we do a little bit conservative filtering. So we remove all the cells with less than 50 transcripts. And if the mitochondrial RNA is too high, we also remove the cells, because the higher mitochondrial read can be indicative of low quality cells, that some RNA degradation has happened during the processing. Overall, for the 3 flow cells we ran, we obtained 3.2 million cells. And it's over 2.5 billion transcripts. It is a pretty impressive number, because for the cell analyzing purpose, when we ran the single cell RNA-seq with the 10x methodology, we usually obtain only 10,000 cells per sample. And we have run so many samples so far. It's about 100 samples from various donors. We are still at 2 million cells profiled. And with this, the Illumina spatial technology, just with 3 flow cells and 12 samples, we are getting 3.2 million cells. So we are getting a lot to digest. So one thing we want to make sure is that the reproducibility of the assay, it is a high throughput assay. So the question is like, how do the replicates they compare? So we compare 2 replicates. One on the left, it is a technical replicates. That's 2 sample from the same block. It's 2 sections. That's profile using the same flow cell. You can see the correlation in terms of the overall gene quantification works very good. And we also tried a neighboring section, but put on a different flow cell. The correlation is also amazing. So it is really very reproducible in terms of the gene quantification. And the biology we are interested in is really how well does this technology deliver the cell types that we want. So in order to do that, we use our single cell nuclear RNA-seq data with which we already have the cell type annotation, and then we can either transfer the label or use this as a basis for annotation. And you can see one spatial map of all the cell types we identify using Illumina's data for the duodenum sample that we have. So it is a healthy sample. You can see the clearly layer, the mucosa, and then the submucosa and the muscle layer. And we see a lot of enterocytes for the small intestine, which are responsible for absorbing the nutrients. And we see the muscle layer, and we can repeat the same analysis for different regions of the intestine. So sigmoid, that's the very last section of the intestine, and you see the mucosa layer, the composition is slightly different compared to the small intestine because the role of the large intestine is mainly for absorbing water and salt. And also the other thing is the muscle layer is much thicker. Overall, we were able to resolve 37 major cell types, both for the small and large intestine that we tested. So I think in our preliminary assessment, the technology works very well. And the other thing, because we use the single nuclear RNA-seq as the basis for annotation, we always want to know how well does it correlate with the single cell nuclear RNA-seq data we also collect on the same sample. Here you can see when we compare the Illumina technology on the x-axis with the single nuclear RNA-seq, the correlation is somewhere between 0.68 and 0.58, depending on which sample we take. So there is a correlation, but it's a not perfect correlation. I think this is expected because the way that we draw the boundaries has influence, and also the technology is a little bit different with the spatial and the single nuclear RNA-seq. You don't get the spatial information at all. And the other thing we did a little bit comparison is how does this compare with the targeted approach, for example, the Xenium from 10x. And you see we apply this Xenium technology to one of our FFPE block, and you see the spatial map. You clearly see the same layering as we saw with the Illumina data. And when we do the correlation analysis, you can see overall for the shared 375 common genes, the Illumina assay agrees pretty well with the Xenium assay for both for FFPE and also for fresh frozen here on the left. So it is a good correlation. And perhaps the correlation is a little bit better when we compare with FFPE, because I think with the fresh frozen, we always get a little bit RNA degradation there. And the other question is, how does it compare in terms of sensitivity? And we are particularly interested in this because with Illumina assay, we are profiling really the whole transcriptome. So it is 37,000 genes after some filtering, and we are looking at 200 million transcripts. And with a targeted assay, the number of genes is only 377, and the number of transcripts is also much lower. But and as expected, the targeted assay with the Xenium comes out a little bit higher in terms of sensitivity. So for the shared genes, it's 375. We are getting about 10 for the Illumina assay and about 15 for the fresh frozen sample, and all the 20 for the FFP sample with the Xenium. And I also show on here on the right for one particular gene, this pan immune marker CD45, that the sensitivity is about similar when we use the fresh frozen sample for the two assays, and the FFP comes a little bit better, at least in our testing with the human intestine. And the other thing we want to know, how does this impact our discovery? So I just subsample the Illumina data to the same 377 genes, and then look at if we can capture the same biology in terms of the spatial map. So here is one example for the duodenum, and we are getting the layers and all the cell types, but without showing you all the cell types, just focus on four major groups, which is the enterocytes. But more importantly, we can also get the important cell type, that's the stem cell at the bottom of the crypt. So these are the cells that's responsible for regenerating our mucosa layer, because we keep losing the mucosa during the digestive process, and the stem cells are there to regenerate them. And it is reassuring that even with the targeted approach, if we subsample the Illumina to the same genes, that we are still recapitulating the exact spatial localization of these stem cells. And maybe more interesting is that the untargeted nature of the Illumina assay allows us to make new discoveries. One thing is really we can identify new markers for enterocytes. Here are two examples. So I'm plotting the two genes. One is the apolipoprotein A1. So this is one enzyme that's responsible for fat digestion in the intestine. And the other one is the adolase B enzyme here on the right. You can clearly see their expression are co-localized with the enterocytes. So, and these are the markers that's usually not included in the pre-designed panel or most panels that you know for the intestine. But because we have the whole transcriptome profiling, we can really see what are the genes that's co-localized with certain cell types and make new discoveries. So in this particular case, we can subgroup or subcluster the enterocytes into two states or two cell types, which are APO1 high and adolase B high, for example. And the other thing is that if you look closer, that there is a little bit layering. So for the adolase B, it's more prominent at the base of the crypt. So if we were to do a gradient analysis, this would pinpoint us to what are the transcripts that's correlated from the crypt to villi axis, for example. And this is ongoing work. And with that, I think I'm at the end of my talk, and here are the brief summaries. So in our testing of the Illumina assay for the human intestine, it really offers very high sensitivity in terms of the transcripts and genes we could profile. And we were very satisfied with the reproducibility of the assay. And when applying the assay for cell alliancing purpose, we could really see all the cell types that we care about for the tissue. And we assessed the assay with how it does it compare with a single nuclear RNA-seq. And we did a little bit of comparison with the Xenium. That's one of the assay we applied in our tissue mapping center. But I think the real value is really to use this as a discovery tool to generate a new hypothesis. I think it's going to be important so that people can find new markers and then they can put them into the targeted panel if they want to follow up. And here are the people who are involved. I really would like to thank Mike and thank you for your attention.